right, welcome to the next section of the echinoderms. So in this section, we will talk about an outdated but still somewhat useful idea about development. And so this is called recapitulation theory, and we can sum it up with the phrase, here, let's type the phrase down here, that, here we go, ontogeny recapitulates phylogeny. Okay, now let's unpack that phrase, because that's three words that don't really mean anything to an outsider on their own, but yet pack in a ton of detail. So ontogeny is the way a thing grows up, from the earliest embryonic stage until it has fully gone through all its metamorphoses and arrived in an adult. Think about that first little ball of cells, and then think about an adult human being, and then think about all the different changes they go through. You've got baby humans that have a tail, that have gills, that have all kinds of crazy structures we do not know from the adults. And together, this entire process is the ontogeny of that organism. So ontogeny, the developmental process. Phylogeny, let's go to the end next. Phylogeny is going to be the phylogenetic tree, which is trying to show us the path that every creature took to get where it is. So we're trying to show how everything that's living connects up together, and at each one of those little branch points, you can imagine a common ancestor that then goes on to found the populations that are the two branches coming out of that point. And so that's the phylogeny. And so the phylogeny would tell you that, for example, chordates like humans and other deuterostomes like echinoids are actually reasonably close ancestors, so much closer than they are to, for example, a protostome, or an ecdysozoan such as an arthropod. But remember all those things, all those the different groups, the nephrozoa, the bilateria, the parahoxozoa. Remember how they all connect up, and eventually you can get to the top of the tree or bottom of the tree, depending on how you think of it. And back there, you've got a last common ancestor of at least all the animals on this particular branch of the tree. So anyway, that's phylogeny. Phylogeny is the family tree. And then recapitulates means, in this one, it's like a recap of an episode on TV, where they just show you all little clips, like a little clip show, of each of the things that happened, and so bam, you're caught up to speed. So the idea is that as a creature develops, it's going to go through different ontogenic or developmental stages. And at each stage, you're going to have a little record of what that creature used to look like, what it was originally. And as you go through all the stages, you're going to basically watch evolution happen as the creature grows up. And so that's the idea. And you can sum it up in these three words: ontogeny recapitulates phylogeny, and it's called recapitulation theory, and it's an older theory, and was proposed as a biological law. It is not held to be strictly true by modern biologists. So what that means is that there are a lot of animals that seem to skip stages. But in general, it's a pretty good guideline. So shown here, you've got, this is from one of the original drawings, and so you've got the early embryonic stages where they all pretty much look the same. And then simpler creatures like fish and salamanders would be earlier branches on the what we call the tetrapod tree. These are these are things, a branch of chordates that are going to have 4 legs. And they're going to start differentiating in their own way at this stage, and everything else stays the same. And then as you would go on, each of these other creatures is going to get off at a different stage. And so something like humans would be highly derived, which means we just go through a lot of different stages. We've been on the planet, or rather our ancestors have been on the planet as long as everything else, because we all share the same common ancestors as every other organism. But we've got a couple more little developmental tricks in there that actually show up in terms of different body parts, different developmental stages, something like that. So that's the idea. What's the evidence? What is the evidence for this? Let's go look, and let's start our text underneath the captions, closed captioning. There we go. Okay, so here are some actual pictures of some creatures, and you can kind of see what's going on. So these are all going to be chordate animals, and it does work pretty well for them, but there are elements of this theory that work well in other animal groups, so it's fairly generally applicable. Something like a lamprey would be a very simple fish. These things don't even have a jaw. And you can see from the early embryo right over here to the intermediate to the late form, it does not change a whole lot. They pretty much start out as a primitive chordate, stay as a primitive chordate, and that's how they do their business. All these other things. So what you're going to look at is the embryo is going to be out here, and quite often the embryo is going to be attached to kind of wrapping around this great big thing. That is a yolk sac. That is made of a bunch of other cells that the little embryo is going to consume for food. And the more yolk sac the parents are able to provide, the longer that embryo can sustain itself before it has to go hunt and acquire food on its own. And so, with a bigger yolk sac, you can get to later developmental stages and do more interesting things. That's the idea. And so, you'll see this sort of gradient, even like a little diagonal gradient, so that an embryonic gar will look like an intermediate stage dogfish, which will look like a late form lamprey, petromyzon, and so on. And as we keep going up here, we just get more and more derived. And so we're just getting to, we're doing a couple more changes in each stage. And we have progressively longer gestation periods. And so the longer gestation allows the embryo to go through more changes and get to a final form. And so you can start out with embryos that are basically the same, from snake to chicken to bat to human, and are even quite similar at this stage, although the snake has started to rapidly multiply the area that's going to eventually become the tail. And then so the snake kind of got off at this stage. It went its own way. Everybody else stayed on track. And then you've got the sort of primitive versions that come out, things like a chicken, and a possum is an Australian animal different from an opossum. And a cat is actually fairly similar to all those others. They resemble some of the primitive mammals. Then you've got really highly derived things that have gone for some crazy extra steps, like bats that develop these sort of leathery wings between their fingers and use them to fly around. And humans, look at us. We're kind of weird looking. Got that big bulbous head up there. Yeah, little baby human. Okay, so where does this all start? Let's go way back to the Cambrian, which, for purposes of this course, let's just say it's a long time ago, about 540 million years ago. This little guy comes from around that time. I think it's closer to about 520 million years ago, if I remember right. And it is called Saccharitis. And Saccharitis is, we believe, a very primitive deuterostome. And what you can see here, it kind of looks like a monster face, right? We don't know which of these things are eyes. Probably these are something like a little sensory pit, or perhaps even where the water goes out. The Saccharitis does have a mouth, and you can see the mouth outlined here, and it does have little plates that seem to go around the mouth. And it's got that nice bilateral symmetry running right up there. This is a kind of a flattened specimen. Here you can see a more three-dimensional specimen that's got a little bit of rock still stuck in its mouth, but they didn't want to clean it out because these are super delicate and they don't want to break them. They actually have to etch these out of rocks with acid. But this is a very primitive look at something very close to the stem group for all deuterostomes. And so here it is, reconstructed in loving 3D and beautiful beige colors. And so here we go. We've got our protostome deuterostome split back here. And so on the protostome side, we got all these guys, and they're going to go on to become the ecdysozoans, and these guys are going to become the trochozoa, etc. Then we got our deuterostomes over here, and they're going to split out into at least three groups. This group, these green guys, are all extinct now. But the earliest branch of that, which is probably pretty close to the branch for all deuterostomes, is this guy, Saccharitis. Based on morphology. If you have questions about that, I can go over it in a little more detail. But I don't think we need to go into all of that right here, or take paleozoology later, and we'll get into it. We'll get right into the weeds. Anyway, two other branches, like sister groups, here would be the chordates. That includes us, we're the vertebrates. and the ambulacrarians, which are the echinoderms. There we go. And a little small group called the enteroneusts. And we can talk about those later. Those come up after. But echinoderms are where we are. And so these are both going to be defined by an ambulacral groove. Ambulacral groove is a little groove that runs along the body, and there can be 1, 2, 3, or 5 of these things. And that groove is going to be lined with tube feet. There are actually going to be pairs of tube feet on either side of the groove. And the groove will be something like a food highway. The tube feet will grab food, and then they will pass it like a bucket brigade putting out a, early 1900s fire. They'll pass it from one foot to another foot to the next foot until it goes all the way into the mouth. And that is going to be the way that they eat. And so that's what echinoderms do. And they're a little different, but we think their common ancestor would have been able to do that as well, at least at some point, maybe right about here. Okay, but within that Echinodermata group, how do we get to the echinoderms that we know and love now? Yeah, interesting. Let's look at the way things have developed. So we think that most, possibly even all, of the echinoderms are going to develop from these groups over here, the pentaradial ones, which means 5-fold symmetric, but maybe not quite. There you can see it's kind of a wonky, twisted, not really 5-fold symmetry. It's just really 5 branches coming off the same highway. Let's see. So we start over here, and some of the very earliest echinoderms are bilaterally symmetric, which kind of makes sense because remember Saccharitis, the weird little monster face on the earlier one? That's also bilaterally symmetric, and it's easier to go in steps. But then look at the way that they develop. And so remembering here that ontogeny is going to recapitulate phylogeny to some extent, leaving out certain steps. I guess that's the asterisk at the end of that. We should expect to see a bilateral stage as the very early stage in embryo development. And that's exactly what we see in every single kind of existing echinoderm. Okay, so the next stage they go through is this kind of asymmetric stage. And this is kind of where what we think is happening with the early metamorphosis. We're going to start to develop symmetry, but we're starting to work on it, basically reorganizing the house. So these are some of these Asturicistis from Asturias in Spain is going to be another one of these little echinoids. And this thing had one little arm, and from trace fossils, we know it would stick this arm into the dirt and then pull itself around. So this is actually the front end. And it's just going to drag the rest of this body around, which is kind of comical when you think about it. The next developmental step, or evolutionary step rather, goes to 3-sided symmetry, so triradial, which is essentially one of these guys, but instead of just having one food groove right along here at the top, or one food groove running up this little feeding arm up here, they're going to have 3 food grooves. And it's just, you can see the fossils actually develop from very young fossils to old ones. Young individuals that were fossilized, rather. And you can see the process that it starts out with one groove, and then it puts in the second and third, just like that. And then in the next step up, you actually put in 2 more. You branch off number 1 and 3. They're each going to split, and so you're going to end up with 5. And that's exactly what we see in the early ones, and that's how we think we get to the modern 5-fold symmetric things. You've got 1, which is like the bottom, then we're going to branch. We're going to have 2 and 3 branch off, and then we're going to have 4 and 5 branch off from 1 and 3. That's it. Lepidocystis goes on, we think, to form the crinoids. Goggia goes on to die out completely. And Stromatocystis either becomes the things that are like starfish, or maybe that is Camptostroma over here, which has a star-like pattern on the top. We think it's inflatable. We think the sides are soft. And so it's like a starfish on top, but that can puff up. Kind of like one of those cushions you might put your feet on next to a fancy couch, but inflatable. Isn't that weird? Yeah, echinoderms are weird. All right, here are some of the early ones, and we're looking at now what's going on the inside. So here's one of the fossils of the early ones. This is one of those Goggia. They're called, what are they called? It's a kind of a crinoid, not pseudocrinoid, not paracrinoid, something like that. Anyway, on the inside of these, every echinoderm is going to have something like a water vascular system. So we have blood, and it runs around in veins and arteries, and that's great. Echinoderms are going to have something similar, a regular vascular system, but they're also going to have a water vascular system, which is a separate set of tubes that are going to pump water around. And they use this to inflate different parts of their body like tube feet. Tube feet are essentially hydraulic. You pull the water out of those and into a little sac at the base of them, and the tube foot deflates like one of those wacky waving inflatable arm tube men. And then you squeeze the water right back in, and bam, you got yourself a nice rigid tube foot again. So they can change their shape in this way and change what different body parts are doing. In addition to that, the water vascular system is going to be there, and it's going to be able to collect some of the waste from the other cells in the body and serve this sort of kidney-like function. And then there's going to be some kind of hole, quite often at the top of the organism, where they are going to exchange and get rid of all the waste that's in the water vascular system. Just dump that out, but keep the water vascular system more or less intact, so like a semi-permeable membrane. On something like an echinoid, this would be a primitive example of something that might go on to become an echinoid. It would be called a madreporite, and it's a little plate on the top that's full of holes, and that is how they get rid of their waste. So that's interesting. Here are some of the early fossils. There you can see one of these little flat guys with a little, basically a little walking arm for dragging itself along. Here's a different one. There's the dragging arm, if you like. This is one that kind of resembles a starfish. They're a thing called goniasteroid starfish that would also have a big row of plates on the outside, 5 little ambulacral grooves that run right on here. And the problem with this one is it's different from a starfish because it doesn't really have a back. These would cement themselves down onto another organism, and so they would never grow a back. They would just live like a little wart stuck to something else. Then you've got guys that have stems, and this guy has 2 arms that come off the top to catch some of that food. Here's one with a little 5-sided groove, and you can see you got your first branch, your second branch, your third branch, and then you got 4 and 5 coming off of those. And so that's how they develop, and so on. Very interesting early forms. But yeah, you can see that echinoderms go through each of these forms as they are developing, leaving out certain steps. So let's go to crinoids first. What's a crinoid? What's an ancient crinoid look like, or a modern crinoid? Well, they have these little arms running right up here, and if you were to get a close-up of the arm, what you can see is that there will be a little groove that goes down the arm, and that is going to be the ambulacral groove. Each of these 5 arms, and sometimes they'll branch out into 10 or even 10 times 2 is. Yeah, 20, I guess. No, 10 times 4, 40 maybe. I don't know. They're going to branch out into even smaller arms, and each one of those is a little conveyor belt that's going to be feeding food into the actual mouth of the crinoid. They're going to have a little ring canal that runs around there so that they can inflate and deflate these things as needed. They're going to have little bony plates that are inside of each one, so that's their little skeleton, because you got to be tough and armored to make it in this world. And their brain is also going to be a ring. Their brain is going to move in a nice little circle, and it seems to be fairly symmetrical. It goes all the way around the body, the brain, so in a little ring, which I think is quite neat. Crinoids, interestingly, I think this is very interesting, are going to develop their skeleton in stages, and each of the stages of their skeleton resembles the skeletons of other kinds of echinoderms. So remember, crinoids are the primitive ones. And so every other echinoderm that is still around today has to come from some version of a modified crinoid. This is the idea, or at least share a common ancestor with a crinoid. So in the early stage, that Doliolaria larva, which is kind of the second stage larva of a crinoid, they're actually going to start to develop some hard parts. And the hard parts are going to start out like this. They start out as a spicule. Wow, where have you seen those before? This one looks like the kind of spicule you would find in a calcarean sponge. One of those 3-sided flat spicules, just like the ones that we saw in the dissection in the lab, like a little Mercedes-Benz symbol missing the circle around the outside. And then as they develop more complicated spicules. So skeletons start from spicules. And that's how everything else kind of connects back to sponges, which is very neat. Just like the family tree would suggest. The more complex spicules up here just show sort of branches on that initial threefold symmetric pattern, or just that sort of random branches coming out. And then they do a neat thing. They just start to branch out and they make kind of a waffle pattern. And if you were to stop right here, that's exactly what we see in holothurians. So those holothurian sea cucumber skeletons consist of little discs and little hooks, which are basically just the embryonic spicules. And they just never develop beyond that stage in terms of their skeletons. The rest of their body keeps developing in interesting ways, but they retain their embryonic versions of the skeleton. And so these are called ossicles, which means like little bones. Think of them as bonelets, yeah, which I think is quite cute. And then, later on, during development, it's going to have a stage where it's just like a little cyst on a stem, and that's going to resemble some of those early forms called cystoids, which are going to look kind of like this guy down here in G. And then it's going to fully undergo another little rearrangement, and you're going to have 5 little arms sprouting off and feeding the crinoid, and this is the pentacrinoid stage. And so at this point, it's become properly a crinoid, and so you see it's still hooked down. So this one has settled and started to cement itself down, and then it's going to grow that little stem using little tiny bones it secretes from right here, and just keep going. And then here are some of the changes that take place in that after we get to that pentacrinoid stage. And it's going to get more complex. We're going to go from the sort of cystidian stage all the way down here to the pentacrinoid, proper late pentacrinoid stage, with the little arms that each branch and have ambulacral grooves down them. And so, their ontogeny, the way they grow up, very much recapitulates their phylogeny. And the coolest thing, which we're going to see when we get to the crinoid section, actually we might see that in another slide, is that there are a lot of crinoids now that don't actually have a stem. But the larvae go through all of these stages. They'll settle down, they'll grow a stem, they'll go through a cystid stage, they'll go through a pentacrinoid stage, and then they will evolve another step. And developmentally, they will cut off this stem and start to move free once they've developed long enough ambulacral grooves on those arms that they can actually sort of use them to flap their way through the water. Very interesting, crinoids. So here's that slide, as I was saying. Here, once again, we've got our little different little embryos. We've got our echinoid embryo at the top. And so this is hours post-fertilization, and these are days post-fertilization. And so you can see the changes that are going on there. It's still fairly conservative. And then at 24 days post-fertilization, it's actually started to branch out a little bit. And these are going to become the primitive spines. This will become the primitive body on those. Shown down here, these are going to be Holothuroidea, so these are our sea cucumbers, which retain essentially an embryonic style morphology. Like very early crinoids, they just never get past that baby stage, and then they go in a completely weird direction from there. It's really neat. And then these are the starfish. They're going to be one of those middle branches, and you can see they're still fairly primitive in their early stages. And then they go on to do their own different thing. So here is a little picture of the Doliolaria larva of a crinoid. And there's the word Doliolaria right there. And this is showing what it becomes. So it's got those little tiny spicules inside. The spicules are going to increase to make little tiny plates. And then the plates are going to become the cup, essentially. So this thing is going to root itself down. This little dot up here is the adhesive pit, not the anus, as I incorrectly said earlier. That's how they're going to stick down to the ground. And then this is going to become the cup. And these little skeletal elements that they've developed inside are going to become the stem. And so they're going to go through this crinoid-like phase. And then they're not going to stop there. So this is the cystid phase. This comes kind of in between those two things. And there you can see the little plates, little embryonic plates looking very, very primitive there. And they're going to go on to develop the pentaradial symmetry, the little pentacrinoid down there. And eventually they are going to developmentally just leave their little stalk behind and swim free. And so the modern swimming crinoids are very much just a next developmental stage from the rooted crinoids, which are the older ones that we find in the fossil record. And their ontogeny does very much recapitulate their phylogeny. Yeah, this is a nice example where that really works out. There are other examples you could point to where it does not work out as well, where it leaves out a lot more steps. But crinoids are wonderfully careful and go through all of those little steps for us. And they're just showing us, they're trying to show us this is where I came from. This is who I am now. This is who I used to be. And yeah, I think that's really fascinating about them. Okay, that's the end of this section of the echinoderms, and we've got many more sections to come, so stay tuned for those. Thank you very much.